Assalamualaikum. Hello, everyone. This is SIL 1001 Cell Biology, the first lecture on cell. Before we begin, let me introduce myself. I am Dr. Nur Ardiana Rajab. You can call me Dr. Ardiana from Genetics and Molecular Biology Program, ISB. So I'm so sorry that there will be only online classes for this semester. And hopefully we are able to make the most of this experience. Okay, so this is me, Dr. Ardiana. Alright, so let's begin. Alright, first let me show you, introduce to you the main reference for this course, which is Cell and Molecular Biology by Gerald Carr. There are actually several other books out there, but this is the book that we are referring to. You may consult me and also other lecturers in this course whether the books that you prefer or you already have, like for example, you can ask our opinion whether it is suitable or not. Okay. Alright, let's move on to our lecture. Cell is the building block of life. All living things is made of cells. Cells, a group of cells, will make tissues, and tissue will make organ, and organ will make system, such as respiratory system and others. If you look at the cell, and if you look closer to it, you can see several structures. Yeah? For example, the nucleus, and inside the nucleus you'll be able to see chromosomes, which are made of DNA. So in this course, we're going to start with looking at how cell theory developed. So we're going to go through a timeline, yeah, looking at discovery by scientists before us, and what and how their contribution led to cell theory. So in this course, you need to, one of the component is for you to understand the cell theory. Okay, I'm not going to read it for you. Okay, so we're going to look at these important event later on in the next few slides. The first one over here, Hans and Zacharias Janssen, these two in 1595, they produced the first compound microscope by combining two convex lenses within the tube. Alright, and then later on, in 1665, we have Robert Hooke. Robert Hooke built thin slices of cork, and he saw network of box-like compartments in what he referred as cells. Yeah, so he used this device to see the thin slices of cork. He's the first to come up with the term of cells. So the word cells actually came from Latin, from cellula, Latin for little rooms. When you look at the structure of the network of box-like of the cork represent the cells, it reminds him of little rooms, or cells. At that time, Hooke devises the compound microscope and illumination system, which was one of the best microscope in his time. However, at that time, he can't see bacteria yet. Not long after that, came Anton van Leeuwenhoek. He was a Dutch textile merchant, but he has a big interest in science. So he produced stronger lenses and in his time the lenses were so powerful that he can produce, or he used 300 times magnification power. Yeah, this is much better compared to the previous one by Robert Hooke, which was just 30 times magnification. He was the first to see and also to describe bacteria, and he was the first to observe living cells, such as the blood, sperm, algae from the pond water. He was the first to observe living cells because previously Robert Hooke, although he saw the network of box-like structure, what we know now by they are all cells, but they are not living things. Yeah, they are already dead cells, because plants, they have a cell wall, and this cell wall can actually remain after the plants are dead. Okay. At that time, he didn't. The people actually not they don't call the observation, I mean what he observed, from our bacteria. He refer them as animalcules, which represent small animals. Yeah, Anton van Leeuwenhoek. So his device actually pretty amazing. It looks like a paddle shape over here, and the size is actually very small compared to the previous one. Is actually bigger, and the lens is actually the size of our the lens of our glasses. It wasn't until the late 1830s that people start to realize the importance of cells, and people start to use compound microscope. What you see over here is the compound microscope that includes the eyepiece and also the objective lenses. Robert Brown, an English botanist, he was the first to observe the nucleus in all plant cells. So what he observed this repeated structure in all of the plant cells that he observed. His German friend Matthias Schleiden importantly concluded that all plant tissues are composed of cells, and that embryonic plant always arises from a single cell. So this conclusion over here is actually very important to produce the first tenet of the cell theory. So I told you about cell theory briefly before. I'm going to explain a bit more soon. Yeah. A year later, German cytologist Theodor Schwann made similar conclusion to animal cells. So all animal tissues are composed of cells, and the embryonic. So I guess this is not suitable. Yeah, only this one. Sorry. So all animal tissues are composed of cell. So in 1839, T. Schwann contributed to the cell theory. And number one, all organism consists of one or more cells, and number two, the cell is the basic structure for all organism. Okay, so it's very important for you to know and understand the cell theory. All right. More than a decade later, they came up with the third principle, or the third tenets of the cell theory, in which cells arose only by the division of other pre-existing cells. Or in other words, cells only develop from pre-existing cells. So this theory was concluded by Rudolf Virchow in 1855. So this slide concludes the cell theory. So you have learned that all living organisms are made of cells, and a cell is the basic unit of life, like the cell theory that I've introduced to you before. And then organisms are either prokaryotes or eukaryotes, and prokaryotes are single cells, and eukaryotes can be single or multi-cells. These 2 points over here, I'm gonna introduce to you very soon, in a bit. And the last one, all cells come from pre-existing cells, and this is actually the third principle of the cell theory. Okay. All right. Next, we're gonna look at different classes of cells. So previously, we've gone through the timeline, the history about how the scientists contributes to the development of the cell theory. And the next important point is to see or to understand the different classes of cells. So cells, basically, you can divide them into the prokaryotes. That includes the bacteria. They are all single cell. And second one is the eukaryote. That includes protists, fungi, plants, and animals. And eukaryotes, they can either be single or multicellular organism. So in the next few slides, what I'm going to show you are different organisms that will represent different classes of cells. The first one is the bacteria. So they are all prokaryotes, unicellular organisms, and bacteria, you can actually divide them based on their shapes. So you see they are coccus, rod, spirillum, and also spirochetes. This is a typical structure of a bacteria. So bacteria, usually the size would be 1 to 2 micrometer or micron. This is flagellum. So you can see flagellum protruding out from the main structure, and the most outer layer is the capsule, the cell wall over here, then you have the cell membrane, and then the cytoplasm. So inside the cytoplasm, you could see this tiny ribosome and also the DNA. Right? So DNA is actually in the cytoplasm, and they are not separated by any structure. This one is mycoplasma. So mycoplasma is actually the smallest living thing on earth, and it lacks the cell wall, unlike the bacteria that we've seen before. So the size of this mycoplasma is 0.1 micrometer. Okay, you have the you can see the cell membrane and then the cytoplasm. Similarly to the bacteria, there's also ribosomes and also DNA. Right? And then this one is cyanobacteria. So cyanobacteria, the size is also very similar to the bacteria. And the different thing about this cyanobacteria is the photosynthetic lamella over here. Quite similar to the bacteria before, we've seen before. There's the capsule, the cell wall, the cell membrane, the DNA, ribosomes. This is the most complex prokaryotes, or complex prokaryotic organism. Now we move on to the eukaryotic organism. So we're going to start with yeast. So yeast, they are a unicellular eukaryotes, what we call or what we refer as a eukaryotic microbe. Then there are also Euglena, the amoeba, Chlamydomonas. I think you are quite, you'll be how to say, these are not very quite common to you guys, and to those who already learn or took biology practical last semester, you might have learned about this. And then there's also simple multicellular organism. An example of the simple multicellular organism is Volvox. So this is how Volvox looks like under the microscope. What's unique about this is that the scientists hypothesized that Volvox originate from Chlamydomonas. So you know Chlamydomonas is a single cell organism. So what happened, what they hypothesized was the Chlamydomonas will, several units of this will form a gonium structure, and gonium structure will then be able to form a multicellular organism, what we call the Volvox. They hypothesized this based on the genomes of these two organisms, which are remarkably very similar. Other examples are of the multicellular organisms are Eulothrix, fungi, and also Hydra. So this Eulothrix is a type of green algae in which the plant body consists of unbranched unicerate filaments. And the filaments, the cells, are actually arranged from one end to another end. About this Hydra, this is quite unique organism. It has the tentacle structure over here that is used to catch its food. And biologists are especially interested in this organism because of their regenerative ability. They do not appear to die of old age, or indeed to age at all. So they can actually die, but probably due to injury, or if they are, being eaten by other organisms. Next are, what I'm going to show. I think you are, some of you might be very familiar with these structures: the mammalian cells, the white blood cells, such as the lymphocyte, the macrophage, and also neutrophils. These cells, they function in the immune system. And there's also red blood cells that are important to carry the oxygen throughout your body. Quite similar to what you've seen before. Addition to that, adipose tissue, the cartilage cells, the bone, and also osteocytes inside the bone. More mammalian cells over here. Epithelial cells, for example, the ciliated columnar epithelial cells of the trachea, the squamous epithelial cells of the skins, and etc., the muscle cells, the nerve cells, and also the specialized cells of the organ. And next, here is a typical animal cell structure. So we're going to start from the most inner structure, which is the nucleolus. Okay, and then here this structure over here is the nuclear envelope, and you can see nuclear pore on the nuclear envelope. And then you would see the I hope you'll be able to recognize this with all of the label. Yeah, most probably in the test or in the exam, I'm going to ask you to label one of this structure. So it's very important for you to know and understand in this lecture. So after the nuclear envelope, you can see structures like the rough endoplasmic reticulum, and then also smooth endoplasmic reticulum. How it's very easy to differentiate them. Their structure, it's very similar, except for the rough endoplasmic reticulum would have ribosomes and the tiny dots attached to it. And then Golgi complex, and then there's also centrioles, and then this one is the mitochondria. There's also free ribosomes in the cytoplasm over here. Yeah. And of course, the vacuole, okay, very small compared to the plant cells, and the most outer layer is the plasma membrane. Let's look at the plant cells. Okay, this is a cross section of from a dicot leaf plants. Yeah, you can see the epidermis, the xylem over here, and this one over here. Yeah, underneath of the plant you can see the gut cells. It's not labeled, but this one is they are actually gut cells. Cross section of stem, you can see epidermis, parenchyma, sclerenchyma, phloem, cambium, the xylem, and also medulla. Let's look at a typical plant cell structure. Okay, not very much different compared to the plant to the animal cells. Yeah, the most significant difference between these two cells is actually the cell wall over here. Okay, of course, and also chloroplast. So similarly, let's start from the most inner part. I wanted to start here. Yeah, again, similarly, like we've seen before, we have the nucleolus, and then the nuclear pore, and then there's rough ER. There's also smooth ER. There's mitochondria. There's also the Golgi apparatus or Golgi complex. In addition to what we've seen in the animal cells, there are also peroxisome, the chloroplast, yeah, and a very big central vacuole. Okay, sometimes plant cells, they can have more than one vacuole. Yeah. And then of course there's a plasma membrane, and then outside of the plasma membrane is the cell wall. Okay, cell wall is very important for the maintaining the shape of the plant cells, because plant cells, they don't have skeleton or bones like us, so they need some extra mechanical strength to support the structure. By now, you already know that cells serve as a basic units of living things, and it's a unit of life in which it able to carry out processes of living. So basic processes, we're going to touch about that in the next slides. And cells is the unit of structure of unicellular organism and also multicellular organism. So multicellular organism needs organ system to carry out functions, and these organs are made of tissues, and tissues are made of cells. And it's also a unit of function for both of the unicellular and also multicellular organism. And you know that cells cannot be synthesized. If you have a mixture of components, let's say for example we have like different components. We have the ribosome, we have the nucleus, we have the vacuole, blah blah blah. We put it in a tube. We cannot synthesize a cell. Yeah, we cannot synthesize a living cells. And the basic processes that I mentioned before, yeah, all living things able to carry out processes of living, for example, the respiration, the nutrition, excretion, movement, sensitivity, growth and repair, and also reproductions. And cells are capable of carrying out process of life because of the presence of the four components. Yeah, these four components are very important to allow the cell to carry out the basic processes. For example, the plasma membrane, the genetic material inside, the nucleus, not nucleus, so depending on whether it is a prokaryotic or eukaryotic, the biosynthetic apparatus, and also metabolic apparatus. So these four components are important for the cells to carry out the processes of life. Now let's look at the four components that we talked about before. Okay, the membrane is a structure that is important to for enclosure, chemical exchange, and also very sensitive to the stimuli. The second one is the genetic material, or DNA. It stores the genetic information, RNA, and also important for RNA and protein synthesis. Yeah, and this DNA is able to replicate, recombine, and also mutate. The third one is the biosynthetic apparatus, for example, the ribosomes for protein synthesis, enzymes, and carbohydrate and lipid biosynthetic pathways. The fourth one, the metabolic apparatus, for the production of energy, enzymes in the prokaryotes, specialized organelles, and the eukaryotes, such as the mitochondria, chloroplast, and also peroxisome. Now, we move on to the differences between the eukaryotic and prokaryotic DNA. So I have introduced to you before different examples of these, the organisms, the eukaryotic and also prokaryotic organisms. I hope you still remember and can recall, or else maybe you can rewind the video and then watch again. But I have made a table over here for you to summarize the differences between the eukaryotic and also prokaryotic cells. The eukaryotic cells, they have membrane-bound nucleus, but not in the prokaryotic cells. And then the second one, DNA complexes with proteins to form chromosome. Okay, in prokaryotic cells, you can see DNA, but the DNA is not associated with proteins, and you cannot see chromosome structure in the prokaryotic cells. The third one is the size of the ribosomes. The eukaryotic cells would use 80S ribosome, while the prokaryotic cells would use 70S ribosomes. Next, we look at the organelle structure. Organelles such as ER, Golgi, mitochondria, and chloroplast can only be observed in the eukaryotic cells, but not in the prokaryotic cells, except for the cyanobacteria, where you can see the photosynthetic lamella. And the next is the presence of cytoskeleton in the eukaryotic cells. Like for example, the mitochondria. You cannot see this in the prokaryotic cells. And then cell divisions. So cell divisions in the eukaryotic cells involve mitosis and meiosis. We're going to learn about this with me in this course. But in the prokaryotic cells, they divide through cell fission. We have learned about the prokaryotic and eukaryotic organism, but there's this one entity, what we call the viruses. The viruses, they are visible under electron microscope, and it has diverse morphology. The most common one is the one with the icosahedral protein capsid, and the viruses, they can have either DNA or RNA as their genetic material, and they are very, very dependent on their hosts, animal, plants, or bacteria, for replication. So without the host, the viruses won't be able to replicate and also propagate. I mentioned before that the viruses are very diverse, in terms of their morphology. This on top, you can see different morphology or different types of DNA viruses that also include the bacteriophage. This is how bacteriophage structure looks like. It has the head over here, the neck, the genetic material, the DNA is inside of the head, and then the tail is over here, and there's also this tail fibers over here. This reminds me of the shape of spider. Yeah. There's also RNA viruses. Yeah, RNA viruses that also include this coronavirus over here. You can see the capsid, the spike protein, the outer layer of the virus. This is how they look like under the microscope. I can easily identify bacteriophage over here, but not really on the others. I'm not the virus expert. So you can see the size. They are very, very small. Yeah. 0.1 micron for the unit. So roughly the size of the virus would be around 25 nanometer to 300 nanometer. What's interesting is whether we can refer viruses as cells, because the most biologists believe that viruses are not living things. Yeah, they don't have plasma membrane, although some with host cell membrane, they don't produce themselves. Again, they are very dependent on the host. There's no metabolic or biosynthetic apparatus in the entity, yeah, in the virus, although some with virus-specific enzyme, and they are dependent on hosts for living processes. So what do we call them? Yeah, so since they are not cells, whether they are living things, whether they are not living things. So I let you ponder about this. Yeah, you can Google for more information. You read more about viruses, and perhaps we can discuss that later. When you study cells, you also need to understand the units, or the diversity of sizes. The smallest would be 1 nanometer, equivalent to 10 angstrom, this symbol over here, and 1 angstrom is actually equivalent to the diameter of a hydrogen atom. Okay, and then 1 micrometer or 1 micron, equivalent to 1000 nanometer, and then 1 millimeter, equivalent to 1000 micrometer, and then 1 meter, equivalent to 1000 millimeter. And then I mentioned before, the virus the smallest would be 25 nanometer and can go up to 300 nanometer. The smallest organism, living thing, is the mycoplasma, which has the size of 0.1 micrometer, whereas the bacteria would be sometimes can go up to 10 micrometer. So cells and organelles are mostly defined by this one, this unit, micrometer or micron. Our eyes, or our naked eyes, can differentiate up to 100 micrometer, and if we take this as a relative power of differentiation, equivalent to 1, the light microscope can be used to differentiate up to 0.25 micrometer, which is 400 times more powerful than our vision. And then if this will make as a relative power of differentiation, electron microscope is much more powerful. It can differentiate up to 0.001 micron, or 250 times more than the light microscope. Okay, that's really powerful. This figure shows the relative size of a number of structures and even organisms. Yeah? So electron microscope can differentiate to the atomic level, and the light microscope over here, we're able to see the chloroplast, the cell structure, and of course much bigger for our human eye. And with these technologies, we are able to understand and study microorganisms and cells. That's the end of my lecture. So I have introduced to you the cell theory and also the prokaryotes and also eukaryotes, as well as viruses. So in the next few lectures, we're going to learn about membranes, the nucleus, intercellular membranes and organelles, the mitochondria, the chloroplast and also Proc system, and we're going to learn about the ribosome components of cytoskeletons, and then finally cell divisions. You're gonna spend 4 weeks with me, and then the next following topics will be continued by Professor Zulkarnain and also Dr. Sharifah, 2 other lecturers in this course. Okay, so if you like to know more about the content of the lecture, please refer to the course info and also proforma files uploaded on Spectrum. If you have any question, please don't hesitate to contact me. Stay safe, everyone. Take care.